Hello everyone, welcome back. We are at the last week of our lecture series for overview and integration of cellular metabolism. In today's class, we are going to discuss two topics. One is alcohol metabolism and diseased condition related to it, and many other is fatty liver. So, the concepts covered in this class will be related to alcohol metabolism. We will learn how metabolism of alcohol occurs in liver and what are their associated enzymes which help in metabolizing alcohol. Then of course, the toxic effect of alcohol, how those toxic effects are imparted, we will learn. Then we will move on to fatty liver, the different causes of fatty liver, the basic biochemical platform which is actually causing fatty liver. Then of course, their metabolic roles, what will be the consequences of fatty liver. So, let us move on to the very first, alcohol metabolism. Now, metabolism of alcohol starts just after ingestion. Remember, for the absorption, alcohol is a molecule, is a small molecule, ethanol mainly is a small molecule, then it is water soluble as well as lipid soluble. So, very fast it is absorbed from oral cavity to stomach to small intestine. The major region, major part is small intestine, but in stomach also, in the gastric mucosa, because you know there is very huge blood supply in the gastric mucosa as well as intestinal mucosa, there is fast passive absorption of alcohol. Now, this absorption basically depends on many other factors, starting from Personalis factor to the substrate which is consumed. There are different factors which actually decides the rate and extent of alcohol absorption, like concentration of the alcohol which is consumed as well as the type of alcoholic beverage. Then regarding the person who is consuming alcohol, their individual factors like age, gender, body weight, their metabolic capacity, then also whether they are fed or not, based on that the rate and how much alcohol will be absorbed are decided. Then definitely after absorption, when it enters in the bloodstream, it is distributed very fast, and the peak blood alcohol concentration, which is known as BAC, it is obtained within 30 to 90 minutes. It is very fast, peak alcohol concentration is achieved after consumption, that too also dependent on those factors I already have mentioned. Now, because it is highly water soluble from blood, it enters the cells. Alcohol freely diffuses across the cell membranes and is distributed to all the cells where there is water and present easily distributed inside the cell. Now, based on that, multiple organs are affected by alcohol, because alcohol can enter very easily. Important organs are brain, liver, heart, kidney, lungs, even skeletal muscles. Now, regarding brain, because alcohol can cross blood brain barrier very easily because of its molecular size, which is very small, as well as it is lipophilic. So, it can impart effects over brain like cognitive function which is hampered, then dementia. These are the things which are hampered when alcohol reaches CNS in different concentrations. Then there are factors which decides the distributions, like how the organ is perfused. So, the blood flow to the organ decides how much alcohol will enter and how it will be distributed. Then again the concentration of the alcohol in the blood and also the rate of metabolism. Remember, after entering the tissue, alcohol is metabolized. So, how fast it is cleared from the system is also determining the effects of alcohol in body. Now, remember, regarding the clearance of alcohol, liver is the most important organ. So, let us move on to metabolism of alcohol in liver. So, this is the major organ where alcohol is metabolized, liver, where there is the enzyme alcohol dehydrogenase. So, what happens basically, alcohol is converted to aldehyde, it is acetaldehyde, and this alcohol dehydrogenase enzyme is present in cytosol. Now, remember one very important thing, when there is conversion of alcohol or ethanol to acetaldehyde, there is conversion of NAD to NADH. So, basically this is one oxidation reaction. Now, what is formed is acetaldehyde, which is the actual toxic metabolite of alcohol. So, it is not ethanol or the end metabolite, rather acetaldehyde, which imparts the main toxic effect of alcohol. Now, 90% of this acetaldehyde is basically taken care of by liver, and the enzyme is acetaldehyde dehydrogenase. Both of them are dehydrogenase. So, definitely both of them are actually using NAD, which is converted to NADH. But importantly, one thing to remember, this NAD is cytosolic. The NAD which is utilized by alcohol dehydrogenase is cytosolic, whereas the NAD which is utilized by acetaldehyde dehydrogenase, that is mitochondrial enzyme. Now, what is formed by acetaldehyde dehydrogenase is acetate, which is a non-toxic product. So, what is the fate of this acetate? Acetate, which is formed in liver, it can be activated to acetyl coenzyme A, and the enzyme is acetyl coenzyme A synthetase. The major isoform is isoform 1, acetyl coenzyme A synthetase 1. Again, that is one cytosolic enzyme. Now, if you remember from the previous classes the fates of acetyl coenzyme A, then you will be able to memorize that the cytosolic acetyl coenzyme A is basically utilized for biosynthesis of cholesterol, fatty acids, like that. Now, this acetyl coenzyme A, which is formed in liver, can enter liver mitochondria to undergo TCA cycle. But the commonest utilization of acetate is basically, acetate is circulated through blood, enters different organs like skeletal muscle, heart, and there it is getting converted to acetyl coenzyme A and utilized as fuel for those organs. So, what happens, the commonest sites are heart or skeletal muscle, where there is once again acetyl coenzyme A synthetase isoform 2, which is the mitochondrial isoform. So, in mitochondria once again acetate is converted to acetyl coenzyme A. Here you can see inside muscle, converted to acetyl coenzyme A, and it takes part in TCA cycle to form to give ATP or produce energy. So, this is the total metabolism of alcohol in liver. The enzymes here important is, first one is alcohol dehydrogenase. Now, this alcohol dehydrogenase, there are different types of isoenzyme based on what type of alcohol it is. So, basically the alcohol the isoforms of alcohol dehydrogenase, they are specific to the chain length of the alcohol. But for ethanol, ethanol is a very small molecule and it has no diverse characteristics. So, what happens, majority, all of the alcohol dehydrogenase, all of the isoforms can metabolize ethanol, but the highest for the highest specificity of the isoform, which is very much specific for ethanol, is the class 1 alcohol dehydrogenase. Now, you can see there are 5 classes of alcohol dehydrogenase. Amongst them there is class 1, which is most specific or very much active for metabolizing alcohol dehydrogenase and abundantly present in liver and also adrenal glands. Now, there are 3 genes for this alcohol dehydrogenase and they together form polymorphic pattern. So, there are different allele of alcohol dehydrogenase which renders polymorphism. So, this is about alcohol dehydrogenase, but for aldehyde dehydrogenase, in the liver the major acetaldehyde dehydrogenase which is present in liver mitochondria is of isoform 2. So, mitochondrial acetaldehyde dehydrogenase is 2, that is also present in heart and skeletal muscle. So, what the mitochondrial isoform is basically aldehyde dehydrogenase isoform 2, whereas the cytosolic isoform is aldehyde dehydrogenase 1. Now, apart from this system, ethanol can be oxidized instead of alcohol dehydrogenase. Ethanol can be oxidized by another system which is known as MEOS, microsomal ethanol oxidizing system. So, definitely the enzyme is basically located in microsome or endoplasmic reticulum. Now, this MEOS or microsomal ethanol oxidizing system is basically a part of cytochrome P450 superfamily, and there are multiple enzymes which can take part in alcohol oxidation, but the most important one is cytochrome P2E1 which is a mixed function oxidase. Now, what is the typical characteristics of this MEOS system where ethanol is converted to acetaldehyde? Here one important thing is molecular oxygen, and another important thing is NADPH. It is not NADH, it is NADPH, which is NADPH and oxygen, they are utilized as electron donor. NADPH is utilized as electron donor and molecular oxygen is the oxidizing agent here. So, this is the difference with alcohol dehydrogenase induced ethanol metabolism. Now, only 10 to 20% of this ethanol oxidation, this type of ethanol oxidation occurs in moderate drinkers. Normally, ethanol is converted to acetaldehyde via alcohol dehydrogenase in liver. Then we are coming to the toxic effects imparted by alcohol and how they are causing this toxicity. So, alcohol metabolism, alcohol toxicity can be of two types. One is acute alcohol toxicity, another is chronic alcohol toxicity. Now, acute alcohol toxicity, the major reason is increased NADH-NAD ratio. As I told you that when alcohol is metabolized, alcohol is converted to acetaldehyde, there is NADH formation, again acetaldehyde is converted to acetate, there is again NADH formation. So, what happens when there is increased amount of alcohol which is metabolized, there is huge amount of NADH formed. Now, what happens when there is a stable metabolic status, metabolic homeostasis in our body, this NADH which is produced can be reoxidized to NAD plus actually this in electron transport chain in mitochondria. But when there is huge amount of alcohol intake which is actually overwhelming the oxidation capacity of those NADH, it causes accumulation of NADH or unavailability of NAD plus. That is the main reason of acute toxicity of ethanol metabolism. Now, you can see here that ethanol when converted to acetaldehyde, cytosolic NADH is increased. Again, acetaldehyde when it is converted to acetate, mitochondrial NADH is also increased. Now, NADH has no rather very poor product inhibition over the enzyme. So, the enzyme goes on and NADH keeps on accumulating. Now, what are the effects rendered by this increased NADH-NAD ratio? Number one is fatty acid metabolism altered. how it is altered? So, there is huge amount of NADH available in mitochondria. Now, what actually beta oxidation of fatty acid does? It provides NADH, and beta oxidation, when it is required in our body, when there is enough energy is not available, ATP is not available. So, basically glucose or carbohydrate is now not available. So, fatty acids needs to provide energy. So, there is beta oxidation, and that beta oxidation actually provides NADH as well as FADH too, which enters, those are the reducing equivalents which enter TCA cycle, which enters electron transport chain to form ATP. Now, because there is enough or adequate amount of NADH in mitochondria, beta oxidation is not occurring, rather inhibited. So, one very important enzyme which is inhibited by high NADH content in cell is CPT1, which is related to beta oxidation of fatty acid. Now, carnitine palmitoyl transferase 1, if you remember, this enzyme was responsible for transferring activated fatty acid from cytosol to mitochondrial matrix. So, basically what happens, this enzyme is inhibited by high NADH, which causes decreased beta oxidation. Now, because there is decreased beta oxidation, so actually there is accumulation of those activated fatty acids. What is the fate of those activated fatty acids? So, what happens, these fatty acyl coenzyme are basically entering re-esterification to form triacylglycerol, and these fatty acyl coenzyme combines with glycerol 3-phosphate. Now, interestingly, synthesis of glycerol 3-phosphate in the glycolysis pathway is also increased, because if you remember once again, dihydroxyacetone phosphate, while it is converted to glycerol 3-phosphate, NADH is required. So, there is adequate or rather high amount of NADH available, which is forming high amount of glycerol 3-phosphate. So, there is fatty acyl coenzyme which is accumulated, there is adequate supply of glycerol 3-phosphate, that is causing formation of triacylglycerol. Now, moreover, this enzyme for esterification of fatty acyl coenzyme is a endoplasmic reticulum located enzyme, which can be easily induced by the concentration of alcohol. So, alcohol can itself induce re-esterification of fatty acid. Now, this triacylglycerol is actually incorporated in VLDL, and VLDL circulates in the blood, which causes hyperlipidemia. So, this is the reason of ethanol induced hyperlipidemia. So, remember why ethanol is causing hyperlipidemia, because beta oxidation is inhibited due to a huge formation of NADH. Those fatty acyl coenzyme along with glycerol 3-phosphate undergoes re-esterification to form triacylglycerol, and that triacylglycerol finally comes to circulation forming VLDL or very low density lipoprotein. Apart from that, these VLDL or triacylglycerol, the triacylglycerol as well as VLDL, when it enters liver, it also provides triacylglycerol to the liver, causing accumulation of triacylglycerol finally in the hepatocytes, causing hepatic steatosis, which means accumulation of liver, and that is also known as fatty liver, which we are going to discuss next. Then these fatty acids, not only they are the effect of this accumulation of fatty acid is not only the effect of inhibited beta oxidation, rather there is increased lipolysis also, because on ethanol consumption there is release of epinephrine, and if you remember once again, epinephrine is the hormone which activates hormone sensitive lipase. Hormone sensitive lipase acts on adipose triacylglycerols, undergoes hydrolysis to release fatty acids in circulation. So, again there is increased influx of fatty acids from lipolysis as well. Also, alcohol induces ketoacidosis. There is excess amount of ketone body formation. Why? Again, you can see acetyl coenzyme, if you remember once again, acetyl coenzyme is the precursor of ketone body formation. Now, there is accumulation of acetyl coenzyme. Why? Number one, because there is adequate availability of NADH, which causes malate formation. So, basically oxaloacetate is actually forming malate. So, oxaloacetate is depleted. Now, because oxaloacetate is forming malate, acetyl coenzyme is not able to react with oxaloacetate to form citrate. So, basically acetyl coenzyme is not able to enter TCA cycle. So, what will be the fate of those acetyl coenzyme? It is diverted to form ketone bodies, excess amount of ketone body is formed. So, that is causing accumulation of ketone body. Moreover, in circulation, ketone body concentration we found in very high amount. Why? You remember, there are few organs which can utilize ketone body as their fuel, but the problem is there is supply of acetate as well. So, acetate actually entering heart, skeletal muscles for production of acetyl coenzyme. So, already there is supply of acetyl coenzyme in the form of acetate to those organs which could have utilized these ketone bodies. So, basically those ketone bodies are only formed, but they are not utilized. So, in blood we are getting very high amount of ketone body, that is causing ketoacidosis. So, remember once again, in alcohol induced ketoacidosis, not only there is increased formation of ketone body, but there is decreased utilization of ketone body as well. So, this is about fatty acid oxidation defects caused by ethanol. Apart from that, there are multiple other toxicities which can be imparted over cell, like lactic acidosis. Once again, that is the cause of increased NADH-NAD ratio. So, lactate is formed in huge amount because there is NADH supply. So, pyruvate is actually forming lactate. Pyruvate that could have formed glucose via neoglucogenesis is actually shifted towards formation of lactate, causing accumulation of lactic acid, lactic acidosis. Now, this accumulated lactate has effect over uric acid excretion also. So, that is causing hyperuricemia. The accumulated lactate actually decreases the excretion of uric acid by kidney. So, remember this point, it often is asked why the patients with gout are advised not to drink excessive amount of alcohol. So, gout is the condition which is already characterized by increased uric acid formation. There is increased uric acid already in our body. Now, if those uric acids are not able to be excreted, these uric acids causes precipitation in different regions, starting from joints to different kidney, like that. So, gout is the condition which can be aggravated if patients take excessive amount of alcohol. Then hypoglycemia. Now, remember, this is very interesting. Patients or the persons, individuals who are on chronic alcoholism, a very frequent drinker, they often forget to take meals. For those patients, hypoglycemia is a very predominant condition. Why? Because remember, when the person is not taking meal, basically their energy supply is dependent on neoglucogenesis. Now, one very important precursor of neoglucogenesis is pyruvate, and amino acids like alanine or lactate itself, they are the neoglucogenetic materials there. Now, due to availability of high amount of NADH, lactate is not able to convert it to pyruvate, rather it is happening the opposite, just the opposite. Pyruvates are actually converted to lactate. Moreover, those pyruvate which are formed from alanine or other neoglucogenetic precursors, they are also forming lactate. So, basically this pathway is actually inhibited and the pyruvate is actually shifted to lactate formation. Moreover, again if you remember oxaloacetate which is present in TCA cycle, those oxaloacetates are also depleted because oxaloacetates are converted to malate. Just in the previous slide I told, because there is availability of NADH in mitochondria, oxaloacetates are basically converted to malate. So, the neoglucogenesis is actually hampered. So, the person is going into hypoglycemia. But interestingly, if a person takes ethanol along with a meal, there is transient hyperglycemia. Hypoglycemia when there is decrease of blood glucose level. Hyperglycemia is when there is increase of blood glucose level. Now, when a person takes alcohol with meal, there is hyperglycemia, though that is a very transient situation. Now, why it is there? Again, if you remember this step, that high NADH actually inhibiting glyceraldehyde 3-phosphate dehydrogenase. So, even if glycerol 3-phosphate is formed, glyceraldehyde 3-phosphate dehydrogenase enzyme is not able to act. So, the TCA cycle is hampered. So, basically glucose is not getting utilized. So, there is transient hyperglycemia in those persons who are taking meal with alcohol. But that is once again is a transient condition. Remember, this is a transient condition. So, we have discussed how the acute toxicity occurs from alcohol. Now, we are coming to chronic ethanol toxicity. Chronic ethanol toxicity is mostly the effect of acetaldehyde, the intermediate, and also free radicals which are generated during alcohol metabolism. Now, the effect of chronic alcoholism are manifested in 3 different forms: fatty liver, alcohol induced hepatitis and cirrhosis. So, fatty liver is the accumulation of lipid in liver which when undergoes inflammation is causing hepatitis, and then finally there is fibrosis and tissue damage in liver which is causing cirrhosis. So, these are the chronic effect of alcohols over our body. Now, remember I am talking about chronic alcoholism. So, the individual is basically taking alcohol very frequently in very high dose for prolonged period. So, apart from the effect of acetaldehyde itself, there is formation of different adducts. So, acetaldehyde which is very highly reactive forms adducts with different amino groups, sulfhydryl groups, nucleotides as well as phospholipids. So, these acetaldehyde adducts are able to impart some toxic effects in our body on chronic accumulation. Now, acetaldehyde, how it causes alcohol induced hepatitis? Generally, acetaldehyde is causing a generalized hepatic protein synthesis decrease. Proteins like calmodulin, ribonuclease, tubulin, these are the very common proteins which their synthesis are hampered. Now, tubulin is one such protein which causes assembly and secretion of VLDL. So, basically there is diminished formation of VLDL. So, what will be the effect? Triacylglycerols which were coming to liver, they are not getting secreted in the circulation, rather accumulated in liver, causing fatty liver. Apart from that, there are multiple other proteins, their synthesis are also hampered, like serum albumin, coagulation factor. Remember, liver is the actually it is the synthetic hub in our body, which is the main site of protein synthesis. So, all the proteins, their synthesis are hampered, namely albumin, coagulation factor, different transport proteins which are actually transferring vitamins, steroids, irons to different organs. Those proteins are accumulated, they are not able to secrete it even, they are also forming adducts with lipid. So, what happens on this accumulation of protein, there is influx of water in hepatocytes causing swelling of the liver, which is finally causing portal hypertension, and finally it is disrupting the whole hepatic cellular architecture. So, this is how alcohol induced hepatitis occurs via acetaldehyde. Next, acetaldehyde adducts, they are also able to impart injury via formation of free radicals. How? You remember glutathione is one very important compound which is responsible for scavenging free radicals in our body. Now, acetaldehyde adducts, they actually inhibits the glutathione function. Also, mitochondria, which is one hub for dealing with the free radicals, its function is also hampered. Function in terms of rate of electron transport hampered, oxidative phosphorylation is hampered, uncoupling of oxidative phosphorylation causing heat release. So, these are the effects over mitochondria. Now, again fatty acid oxidation, it occurs inside mitochondria. So, whenever there is mitochondrial function hampered, fatty acid oxidation will also be hampered. It will cause more lipid accumulation. So, basically this toxicity is occurring in a cycle. Those fatty acid attacks with lipid, they are hampering the mitochondrial function, then hampering the beta oxidation of fatty acid, causing more accumulation of lipid. Those lipids are once again forming adduct with acetaldehyde. So, this is a progressive cycle of damage due to free radical formation. Then one very important enzyme here, cytochrome P2E1, which is important in Meio's system for metabolizing alcohol. This is one very important site of free radical formation. Enzymes which contain FMN, FAD in reductase, flavin adenine dinucleotide, flavin mononucleotide, or present in reductase or in heme of cytochrome P450 system, they actually transfer single electrons, and they are the very soft targets where free radical is formed. Now, one such free radical is hydroxyethyl radical, which is accumulated during chronic ethanol abuse. So, basically these enzymes are actually induced by alcohol to form more, because these enzymes, remember these are the microsomal enzymes, and microsomal enzymes are very much inducible by the substrate. So, whenever there is increased amount of substrate alcohol, those enzymes are their activity is actually increased, and because their activity is increased, the free radical generation is also increased. Now, one very important target of free radical induced damage is phospholipids, which are present in membrane. Now, just imagine mitochondrial membrane is basically affected by the lipid peroxides, the free radicals formed during alcohol metabolism. Now, on this lipid peroxidation, enzymes which are present over mitochondria or the electron transport chain, that will be hampered, as well as if there is leakage in mitochondrial membrane, there will be uncoupling of the whole electron transport chain, finally causing an energy disbalance as well as tissue damage. Also, there are different types of carcinogens. Carcinogens are those products which can cause cancer in our body. So, but they remain in inactive form. There should be some trigger which activates these carcinogens. So, free radicals are such triggers which can activate carcinogens in liver, causing hepatocellular carcinoma. So, this is one cause of hepatocellular carcinoma induced by alcohol, chronic alcoholism. So, now we are coming to our next topic, that is fatty liver. The spectrum is also known as fatty liver disease or hepatosteatosis. Now, fatty liver is basically the accumulation of lipid in liver, but remember, liver is the site of lipid biosynthesis. In a well fed person, these are the metabolic steps related to lipid metabolism which occurs in liver, like de novo synthesis of fatty acids, triacylglycerol synthesis, cholesterol synthesis, phospholipid synthesis, glycolipid biosynthesis, VLDL biosynthesis. All these metabolism or biosynthesis of lipid molecule actually occurs in liver, but liver is not the organ which stores lipid. Normally less than 5%, 3 to 5% only lipids are present in hepatocytes. Majority of the lipids which are actually synthesized in liver, they are released in the circulation in the form of VLDL. Now, the problem is, if there is increased supply of lipid, production of lipid or supply of lipid in liver, which cannot be at pace with its secretion in the form of VLDL, or the supply of lipid is okay, but there is some problem with VLDL formation, this condition can lead to accumulation of lipid in liver, causing fatty liver. So, basically fatty liver is steatosis, where more than 5% of lipid is actually retained in hepatocyte. Now, remember, fatty liver disease is basically a reversible condition. Large vacuoles of lipid accumulates in liver, but if it undergoes, if it remains for long standing, for long duration, it can have inflammation. So, this progressive inflammation over steatosis is actually causing steatohepatitis. What will be the consequences of this? There will be non-alcoholic fatty liver disease. Remember, alcohol can cause the spectrum of fatty liver disease, but there are other triggers apart from alcohol that also can cause fatty liver or the spectrum. So, that can be non-alcoholic fatty liver disease, non-alcoholic steatohepatitis, cirrhosis, hepatocellular carcinoma, finally liver failure. The whole organization of liver functions are hampered, causing liver failure, starting from synthesis to excretion, everything. So, what are the metabolic defect actually which is finally developing fatty liver? So, number one is, if there is the main point of the fatty liver formation is definitely there is an imbalance between supply and mobilization. So, supply in the form of endogenous triacylglycerol formation and mobilization in the form of VLDL formation. So, whatever triacylglycerol is actually formed or reaches liver, those are released via VLDL instead of accumulating. But if these balance is hampered, there is accumulation. Now, what are the causes which can cause triacylglycerol formation defect? If there is increased flux of fatty acid in liver, or if the diet which can form increased free fatty acid in liver, there will be huge amount of triacyl glycerol or substrate supply. Again, conditions where triacyl glycerol formation is fine, but their mobilization or release in the form of VLDL is defective, that can cause fatty liver, like defective apoprotein synthesis. Remember, apoprotein is one very important protein part for lipoprotein formation. The lipid which is assembled in lipoprotein, their synthesis can be hampered. Synthesis of apoprotein and lipid are fine, but their assembly is defective. Then finally, the assembly is okay, but the formed VLDL is not able to be secreted. So, these are the defects which can lead to fatty liver. Let us see more in details. So, what are the increased cause of free fatty acid influx? If there is increased lipolysis from adipose tissue, that can cause increased free fatty acid flux. The condition can be uncontrolled diabetes mellitus or prolonged starvation, where there is increased lipolysis, which I have already discussed in integration of metabolism. Then diets which contain very high fat or high carbohydrate, carbohydrate can form fat in liver, and those can be the influx for TAG or triacylglycerol formation. Then defects in VLDL formation, apoprotein synthesis can be defective. Now, there are multiple drugs which inhibits protein synthesis, like puromycin, ethionine. Now, ethionine, if you hear this, it is quite resembling with methionine. So, basically it is actually causing some problem in methionine metabolism. What is the problem? Remember, when there is this ethionine is have some structural resemblance with methionine. So, instead of methionine, while there is S-adenosyl methionine formation, ethionine can be incorporated instead of methionine. Now, what happens? Adenosyl group, which has adenine, it is basically blocked, because finally when there is ethionine instead of methionine, that cannot take part in methyl transfer reaction. So, basically adenine is trapped. Now, when there is adenine trapped, there is definitely some problem in ATP formation. So, basically that is causing energy deficient status, which is induced by ethionine. Again, CCl4, chloroform, lead, these are the molecules which inhibits protein synthesis, as well as a generalized malnutrition, protein energy malnutrition, that can also cause decreased protein availability on synthesis, which actually causes apoprotein synthesis. It causes defective apoprotein synthesis, which is finally reflected in VLDL formation. Then there is defective lipid synthesis. So, phospholipid synthesis can be hampered, because there are different molecules which are responsible for phospholipid synthesis. Those are known as lipotropic factors. So, if there is deficiency of lipotropic factors or defect in lipotropic factors, they can cause defective lipid synthesis, which is reflected in VLDL formation defect. Then the assembly of VLDL can also be defective. Remember, if there is damage in hepatocytes like hepatitis, alcohol induced hepatitis, or some drug induced hepatitis, viral hepatitis, infective or inflammatory conditions, if the cell which is actually causing the assembly, hepatocyte, is damaged, the ultimate formation of lipoprotein will be hampered. Then secretory defect in VLDL. Now, orotic acid is one such molecule which causes secretory defect in VLDL. Actually, VLDL undergoes glycosylation for its secretion. Now, orotic acid inhibits this glycosylation on VLDL. So, VLDL ultimately which is formed is only residing in liver, is not able to be secreted in the circulation. So, these are the conditions where mobilization of fat is hampered in fatty liver. Now, one very important term I have told, that is lipotropic factors, which is that can be defined as the substances which help in formation of phospholipid, and because they help in formation of phospholipid, they are very important in mobilization of VLDL from liver. Now, what are the molecules or what are the lipotropic factors? Now, if you remember from the phospholipid synthesis class, choline, methionine, serine, these are the molecules which are important for synthesizing phospholipid. Choline for lecithin, phosphatidyl choline. Methionine is the methyl group donor S adenosyl methionine to form phospholipid. Again, phosphatidyl serine is also one very important phospholipid. So, these are, if these are deficient, adequate phospholipid cannot be formed. Then essential fatty acids like linoleic acid, they are the part of phospholipid. Then vitamin E and selenium. Now, these are the molecules which are important for free radical scavenging. So, free radical induced damage causes phospholipid formation defect. So, these are the lipotropic factors which are important for phospholipid formation, and their deficiency can cause defective VLDL formation. So, finally, fatty liver, remember this is the imbalance between the triacylglycerol formation and its mobilization. So, let us learn the key points of this session. We have discussed alcohol metabolism. The major site of alcohol metabolism is in liver, where hepatic alcohol dehydrogenase, which is the cytosolic enzyme, and acetaldehyde dehydrogenase, which is the mitochondrial enzyme, they are the most important enzymes. Now, the toxic intermediate of alcohol metabolism is acetaldehyde. Apart from this system, there is MEOS, microsomal oxidizing system, which is responsible for 10 to 20% of ingested ethanol metabolism, and the acute alcohol toxicity we have discussed, that is where the main reason of acute toxicity is basically increased NADH NAD ratio in liver. And finally, the next topic, fatty liver, we have discussed regarding the pathogenesis, how the imbalance of influx of lipid and clearance are actually causing fatty liver, and also discuss lipotropic factors. These are my references. Thank you all.